welcome everyone and welcome Matthias, and we're looking forward to your very interesting topic.
Yeah, thank you for joining today, and thanks to the ICPS for allowing me to talk about some of my favorite topics, which is Nepenthes and the sex in plants. So I'm going to start with a little bit of background about sex in plants. And usually plants are hermaphrodites. So they make both male and female organs on the same plant, as you can see here on the left. And that's the case for more than 90% of all flowering plants. But a couple of them they do it differently. So some of those 90% make separate male and female flowers in the same plant. As you can see in this diagram here. So on different parts of the plant, you would have male flowers, and on other parts, female flowers. And then there are the small minority of plants that actually have separate male and female individuals. And that condition is called dioecy. So what makes this really interesting to study is that in plants we have this diversity in how sex is expressed, and we can ask questions about why and how an individual should be either male or female or both. And that's something that's much harder to do in animals, because most animals are just male or female, quite boring. And in plants, we have this great diversity and many independent times of evolution, mostly going from this hermaphrodite to separate male and female dioecious conditions. So but this talk today is about Nepenthes. So as most of you probably know, Nepenthes is quite a big genus. So I've written here 140 species or so in the core region, which is now outdated. There are already more than 160, maybe more than 180 described names from the genus in Southeast Asia, maybe here in Indonesia and Malaysia, in the Philippines, and a lot of species also in Indochina. Then we've got a couple of outlying species in Madagascar, Seychelles, Sri Lanka, and northeastern India, and New Caledonia in the Pacific, and some other Pacific islands also have populations. So this is what we're dealing with. But specifically today about sex in Nepenthes. So I'm going to start this by reviewing the Nepenthes life cycle. So a Nepenthes plant, all Nepenthes plants start their lives as one of these tiny seeds that you can see here falling out from the mother plant's capsules. Very small, very light, very little resources to establish the seedling. But eventually, after a number of years, first they germinate, then they look like this. So this is actually a picture from the ICPS website. One of the nicest pictures of Nepenthes seedlings I could find. So here you see, just after germination, the first true leaf that they make is already a carnivorous organ. But these look very different from Nepenthes plants later in their life. Then after the seedling stage, typically they go to a rosette stage, where they make the so-called rosette pitches or lower pitches. So they are characterized by short internodes. So this is the plant doesn't climb; it's just growing near the ground. So the tendril of these lower pitches joins. So here we see the rosette pitchers of a Nepenthes. So early in the life cycle, it just grows near the ground, and the pitchers are inserted with the leaf. So the tendril is facing away from the lid insertion. So the opening of the pitcher is facing towards the stem. And then later in life, it will start climbing, and the pitches are totally different, where they the tendril joins the pitcher from the back, so at the same side where the lid is. And then eventually, when the plant is big enough, it will decide to flower. So it can happen even in very small plants like this one. Complete Nepenthes gracillis has made upper pitches, as you can see here, inserted from the back, the tendril actually, but near the ground. So it also sometimes happens, and it started to put out a flower. But usually the plants grow very big and climb over bushes until they throw out flowers, like you can see here. Or even they climb up many meters high into the trees, as in this Nepenthes rhombicaulus, and a huge vegetative mass, and just making a few flower spikes at the top. And then the plant lives for many years, and it can flower repeatedly and repeatedly. Another species that flowers in the climbing stage is here, Nepenthes charniana, massive flower spikes on huge plants. And other species actually flower from the rosette. So those that grow in open habitats, where there are not many trees to climb around, such as this Nepenthes attenboroughi, they actually make upper pitches, but they sit near the ground, and they flower from a rosette habit. Similarly, this is Nepenthes argentii, tiny plant, which still makes a long flower spike and probably costs a lot of energy to make. As you can see here, the new leaves on this plant are, that come out just after the inflorescence are much smaller, indicating that it really puts a lot of power into this inflorescence. Yeah, but what we have here is males and females, as you probably noticed before. So this is from the same species. This is Nepenthes lamii in Papua New Guinea, and here's a male inflorescence on the left and the female inflorescence on the right. To show you a bit more details about these inflorescences, it's a close-up of male flower buds from a different species. And male flower buds in all species typically tend to be more or less round. So if your Nepenthes is coming into flower and you're wondering, is it a boy or a girl? First thing is to look at the roundness of the flower buds, because in the female, the flower buds here tend to be more or less oval shaped, so more long. And this is usually a way to tell a bit before it's flowering which sex it may have. A bit of a closer look at the flowers themselves. So this is a beautiful drawing of an individual male flower separated from the spike. And as you can see, it has petals here. So it doesn't have a calyx and petals like a normal flower, but it has only tepals. So there's only one ring of organs around the actual sex organs, which you can see here in the middle. And this is basically a fused clump of what in other flowers are many individual male organs that is just fused together in the Nepenthes to one thing. Here showing some of the internal vasculature, and here cut up in half, the cross section. You can see this little head here, and on the surface of this head are what's called the anthers. So these are the actual organs that produce the pollen. And the pollen in Nepenthes is also quite special for plants, because pollen grains are not individually released, but they are released in groups of 4. So there's always 4 pollen grains stick together as one grain, a super grain sort of. So it's called a pollen tetrad. So you don't have to put quite as many pollen grains on your female Nepenthes as you think, because each one grain that you put can actually fertilize 4, can actually make 4 seed. So the female flower looks completely different. There's no trace of any male organs, and so the outer parts look similar, the tepals here, so the single whorl of enveloping leaves, and in the middle is the ovary and the stigma on top. So the stigma is the surface where the pollen would land and germinate and grow to fertilize the the ovules. You can see it here in the cross section on top. Pollen grains can land, and then they grow through this tissue and fertilize the ovules here in this ovary. And this is another cross section in the other dimension, where you can see that so there are hollow chambers inside with the premature seed that can grow here and take space later. Okay, but how does Nepenthes mating actually work? So very little is known about the pollination ecology of Nepenthes. What is clear, though, is that they are pollinated by insects. There was some argument whether it could be pollinated by wind, but it turns out that the pollen grains that they make are quite too large to be transported by wind. And one study has actually, one or two studies only have done the experiment, basically wrapping Nepenthes flowers with some mesh in the wild to exclude any animals and then see the seed set. And it turns out then you don't get any seed. Yeah, so they really need animals to visit the flowers, such as this fly here on a Nepenthes fipiata. And the insects come to visit these flowers, because of the nectar glands on the tepals. Yeah, and they look very similar in both male and female, and so the flower scent in Nepenthes is also special. It's very different from the scent of the pitchers, importantly, because otherwise the plant might eat its own pollinators. It's definitely a bad idea. So some of you know the typical smell of a Nepenthes flower, and it's can be quite annoying in small spaces, but it's definitely strong, and that is also an indication that they want to attract insects to move their pollen. Okay. But what is also interesting about the flower biology is possible sexual dimorphism. So in this example again, Nepenthes lamia, it exemplifies it quite well. If you count how many flowers the male makes on an inflorescence and how many the female makes, there's quite a big difference. So males typically tend to make, so in the literature, about 1.6 to almost 6 times more flowers than the female from the same species. And why is that? Why would they do that? And so this is some data that I collected myself, and the reason might have something to do with how expensive these flowers are for the plant to make. So this is the dry weight of flowers. So after flowering, I collected the inflorescences and just weighed how heavy are the individual flowers. And male flowers, they're much lighter here in green, and in red we see a few data points for female flowers. They tend to be about 10 times heavier than the individual male flowers. So, and this is not counting the seed that the female had also to make. Yeah, so it looks like here the male flowers are cheaper to make than the female flowers, but we cannot really be sure because the male also has to make the pollen, and the pollen can be very expensive because it uses a lot of phosphorus that the plants have to catch from their prey. When we look at the level of the whole inflorescence, this pattern is repeated. So the total inflorescence of females is also much heavier than the inflorescence of the male. So even though the male makes more flowers, still the dry weight that the female has to support is still higher for this Nepenthes gracilis case. Okay. Yeah, you probably also observed that in your own plants, females make many fewer flowers per flower stalk than males. Yeah, but then there are other aspects of male and female Nepenthes that might be different, and this is one of the very few data sets that exists about that. And so what these researchers here did quite a while ago, they counted the male and female plants in 2 different habitats for Nepenthes gracilis, and in an open dry habitat where the forest had been cut, most plants are male and very few females. In gracilis and in Rafflesiana, there were even only males in this habitat. Whereas if you walk a few meters away into the shaded forest, the sex ratio is reversed. Then in the shaded forest, there are much more females than males in both species. So that indicates that male and female do have some kind of different physiology, but we know very little about this and why that is the case. So typically, if you imagine that seed is just thrown at random into both of these habitats and the plants grow, and they have 50-50 sex ratio to begin with, then to get this result, one would have to assume that one of the sexes dies off more than the other. So that perhaps the open habitat is more stressful and therefore the females die earlier than the males. But how that is actually connected to their reproduction and their being male or female is totally unknown. Okay, then there's something to say about Nepenthes sex beyond the species, because Nepenthes often grow with 2 or more species together in the same place, and often their flowering times also overlap. It's another topic about which quite little is known, when they flower in the wild and whether the different species have different flowering times. But here's an example where I found a Nepenthes mirabilis habitat that was also a Nepenthes rafflesiana habitat. And among them, you can find these F1 hybrid plants that I put here in the middle. Yeah, so how is that possible? Why can Nepenthes do that? And what's the meaning of that? So we would think that if now this F1 hybrid crosses back with its parents, and that progeny crosses back again and again, that over time, mirabilis and rafflesiana might just blend into one hybrid swarm. But that is not the case. So this is a fascinating question that I've thought about a lot, but I haven't been able to answer. So something is preventing them from just blending together. But why can they do that? And part of the answer is that Nepenthes are a quite young genus. So here is a phylogenetic tree depicting how we think Nepenthes are related to each other and to other species. And although the Nepenthes lineage separated from others some 76 million years ago or so, like way back in the Cretaceous or earlier, the species that we now have, they're quite closely related. They're at most about 20 million years separated from another, like as you can see here. And most of the species in South Asia, they're probably on the order of only 5 million years separated from another, and many of them are even less. So they're not very different in their genomes, and that definitely would help to make hybrids and let the hybrids also be fertile. Nepenthes have also hybridized a lot in their past. So here's a different version of that phylogenetic tree where I inferred introgression events, so hybridization in the past. And there are a lot of these events between different Nepenthes lineages. Here's a paper where I describe more of that issue. But just one of the examples here, so the Montanais, as they are named here, and the Pyrophytae, those are the Indochina species and the Sumatra species. It seems that these are actually at their very deep ancestry, these originated from hybridization between something that was related to Nepenthes mapoluensis and something more ancient. Yeah, that coexisted at the time, came together, and then formed the basis of what is now the Sumatra and Indochina lineages of Nepenthes. And there are lots of other events like this in that radiation. So hybridization is important and has been ongoing for a long time in Nepenthes and hasn't stopped them from becoming many different species anyway. Okay, now coming to how sex is actually determined in Nepenthes. How does it work? So nature has invented lots of different ways to make males and females. First thing what I already talked about is the hermaphrodites, so organisms, plants, but also some animals. They can be both male and female at the same time. And in some, it's also like the case that they start out as one sex, and then over time, as they age, they change sex and become go from male to female or the other way around. Then there is environmental sex determination, famous in many reptiles, like turtles here, where the temperature at which the eggs are raised determines whether the individual is a male or a female. There are even other organisms where social factors determine what sex they become. So when there are many males around, then the individual would more likely be choosing to become a female, and the other way around. And then there's genotypic sex determination. That's the same as in our species. So where we have sex chromosomes that determine genetic factor, whether one is a male or a female. There are XY sex chromosome systems like the human one, and there are ZW systems like in birds, where the females are actually the one that carry the odd chromosome. And there are even more confusing genetic sex determination systems where there's not just one pair of sex chromosomes, but there are multiple independent genetic factors that determine whether the individual is male or female. And in mosses, okay, we don't have to talk about the mosses now, but they have also sex chromosomes that are called U and V sex chromosomes, but that work differently from both ZW and XY. So, but what's the story in Nepenthes? Sex chromosomes can be very obvious, like this human karyotype shows. So these are the human 22 plus chromosomes laid out and sorted. So that 22 pairs, and the 23rd is not really a pair. That's the X and Y chromosome. So this would have been a male. And they can also be quite obvious in plants. So this is one of these dioecious plants, the white campion. It's invasive in the US, I think. And this one has XY sex chromosomes that can be distinguished just by the size. So if these are stained and you look at them through a microscope, then you can see there's a small X chromosome and a giant Y chromosome that only occurs in plants that have male flowers only. But in Nepenthes, the chromosomes all look very similar. And as Andreas Vistuva and someone from the University of Munich found out already in the 90s, we cannot really tell any difference between the species or between the sexes in Nepenthes chromosomes by looking at them under an optical microscope. That looked like Nepenthes doesn't have genetic sex determination. But I wondered, is that really the case? And so during my PhD, I looked closer by sequencing genomes. So this is here a chromosome sketched. But when you look closer and closer and closer into a chromosome, then you can unravel the actual strand of DNA. And by modern technology, we can determine the sequence of the base pairs in these DNA strands. So if we look very closely, maybe there's something in Nepenthes. And it turns out, yes. So this is from Nepenthes Genome Project that Kenji Fukushima initiated a few years ago and where I was fortunate to collaborate. So here we scanned all the 40 chromosomes in Nepenthes gracilis for possible genetic determinators of sex. So I spare you the statistics here, but the essence is, so we have laid out 1 to 40 chromosomes, each neighboring chromosome distinguished by color, and gray is basically noise or something we cannot tell. But here, the higher the points on the axis, on the y-axis, on the vertical axis, the more significant is the association with sex. And so in chromosome 20, right about the center, there is a clear signal that distinguishes males and females. If we look even closer into that part of the genome, we see that it's quite small, this region that is elevated in that statistic. And even closer, here now we can see that this region is about 2 no, just about 1.5 million base pairs long. That is a large number, but it's not very much in terms of the genome. The whole genome is 800 million base pairs long. So this is just 1.5 million. And in this region, that is the Y chromosome, there are 3 important genes marked here in red. So what are these 3 genes about? Here we see how these genes are active in developing flowers. And 3 of them are only active in male developing flowers, not in the flowers of females. So this is the MMD one, the DID one, and the LFYY. So you can see, so blue is gene activity in males, and orange is gene activity in females. So these have something to do with making the flower male or female, whereas this 4th one is not. So that has similar activity in both sexes. So one of these Y chromosome genes is this DID one that I mentioned before, and that was the first one that I discovered that distinguishes male and female Nepenthes. And this gene is very interesting because we think that it has something to do with pollen development. We do not know what this gene actually does in Nepenthes, but we know what this gene does in other plants. So other plants have also copies of this gene. And if that copy is deleted, then the plant is male sterile. So then it becomes a female. As you can see here. So this is a healthy pollen producing on the left and not producing any real pollen on the right. Okay, so female Nepenthes lack this gene. That means they cannot make pollen that works, very likely. Yeah, and with the help of this gene, we can also no, actually, I want to go back and show you something more about the other gene. So this other gene that is, so the other 2 genes, so this MMD one, it has a similar function to this DID one that I just explained. But the 3rd one is very different. The 3rd one, this LFYY, is probably the main sex determination gene in Nepenthes, because we know that in other plants, this gene determines the identity of floral organs. So this gene could, in principle, so that's the hypothesis, turn the whole development of the flower towards only male or only female. And that, because it is the only gene of this kind in the Y chromosome, it is very likely that this gene is actually doing that. But experiments need to be done to confirm it. Okay, so but with this dysfunctional tapetum gene that was discovered first, we can actually do molecular sexing of Nepenthes. And that is very helpful because breeders and also conservationists often want to know the sex of plants before they flower. And that is possible with the help of PCR. So we can, PCR is polymerase chain reaction, standard technique in molecular biology that helps to amplify the number of copies of any particular sequence from a genome. And we need to do that because individual genomes are not visible. But with PCR, one can amplify individual pieces. And typically one goes for about, 30 cycles of PCR, so that each copy that exists in the beginning, in the end, exists more than 1 billion times. And this helps us to visualize these pieces of DNA. With the help of specific primers, which make the amplification target specific. So I made primers for this dysfunctional tapetum gene in Nepenthes, and this is the result of the PCR reaction from different male and female individuals that I had at the time. And here, this is electrophoresis. And it essentially what comes down to that males always have this white spot here, and females don't. So in alba marginata, the male had it, 3 females not, or almost nothing. In ampullaria, okay, this one ampullaria failed. the control actually. But then the male had it and the females have something else. In Bicalcarata, similar. In Gracilis, the 3 males have it, the 3 females don't. In Hemsleyana, 2 males have it, 2 females don't. And in Mirabilis, I had only 2 males, but they both had it. So this is molecular sexing of Nepenthes, and that is applied by some companies like Carnivorous has explored that, and other people also offer this as a service, I think. Then another question that many of you may be wondering about is whether can Nepenthes change sex? And so what I think is the following. So this found out that the Y chromosome has these 3 important genes on it, 2 of which are essential for pollen development. And the 3rd one probably turns off the female development and enforces that the flower becomes wholly male. So that means the X chromosomes don't have any of these 3 genes, and therefore females, they lack the 2 genes that are essential for pollen production. And so I think there is no hope that genetic female can ever become, can ever make good pollen. But the situation is different for the males. So the males have the full set of genes, so the full set of genes to make female function and male function. So they could, in principle, make it. So maybe they can. And maybe there exists a method that has still to be discovered. So the prize is out. Who can find the correct treatment to turn male Nepenthes into females, or at least let them make some female flowers. So this is standard practice, standard idea in cannabis breeders, for example. They induce male flowers by spraying female plants with gibberellins, which are plant hormones. So if you have a lot of flowering Nepenthes, please spray them with all kinds of hormones to find what changes their sex. Yeah, so there are many open questions about Nepenthes sex. So what triggers flowering is not really known. Is there any sex-specific selection in sexual dimorphism beyond the flowers? And what is it about? Which factors actually preclude this blending of species in the wild? So what is reproductive isolation about? And how do these specific genes that we have discovered actually lead to the unisexual flower development? Yeah, and with that, I'm hoping for lots of questions and comments. Yeah, please reach out to me if you have any thoughts. I'm always happy to discuss Nepenthes. Thank you.
Thank you for attending another great ICPS webinar. My name is Kenny Coogan. I'm the Education Director and board member for the ICPS, and we are a group of horticulturists and hobbyists and scientists from all over the world. And we've been around for about 50 years, and we've evolved into a truly international society, and we offer a professional quality journal. We have a lot of free webinars coming up, so you can check out our schedule by visiting our Facebook page or by going to our official membership page, which is Club Express. You see that all of these webinars are between now, the end of October, through the beginning of January, and then I'll be scheduling more webinars for the early spring. You can reach out to me at kenny@carnivorousplants.org if you want to be a future presenter for the ICPS. You could have a doctorate in plants, or you could be somebody who grows a lot of plants in your backyard. We're looking for all different types of expertise. A couple of years ago, I created 5 animated videos, and these are great for people who are just getting into growing carnivorous plants, or if you are a teacher, or if you have young kids, we encourage you to utilize our videos. We have videos on what is a carnivorous plant, how to grow carnivorous plants indoors, how to grow them outdoors, how to hand feed a Venus flytrap, and how to acclimate carnivorous plants. Because as you know, it's a little difficult to get carnivorous plants locally, so you might have to end up getting them shipped to your house. 4 or 5 years ago, somebody, a member, recommended that we have a World Carnivorous Plant Day. So now in 2025, we'll have our 5th annual World Carnivorous Plant Day, which is always the first Wednesday of May. So the date changes, but it's always the first Wednesday of May. If you go to our YouTube channel, you can see all of our past videos where we celebrated carnivorous plants. This is the website that I mentioned earlier about seeing the schedule for webinars. If you become a member, you get lots of great benefits. One of them is you get to support carnivorous plant conservation and education. You also get access to our seed bank, which we oftentimes can ship internationally. And best part of being a member is that you get access to our carnivorous plant newsletter, which features regular columns about horticulture. It includes research papers, field trip reports, conservation news, book and literature reviews, communication from members. This is the official place to register a cultivar. A cultivar is a cultivated variety. So if you are breeding things and you find something really special and you want to name it, we are the official location for all carnivorous plants. You can also publish artwork, meeting announcements. So if you're in Germany and you're part of the German-speaking carnivorous plant society and you want to mention like a really big event, you can publish it in our CPN. And another fun thing that I started a couple years ago was the Carnivores in the Classroom Grant. This happens every August, the month of August, and we encourage teachers from all over the world to apply. Teachers are going to, or students will make a small PowerPoint presentation on how they will incorporate carnivorous plants into their classroom, and the winners will receive $150 worth of live carnivorous plants to their classroom, shipped for free. You can learn more about that by going to carnivorousplants.org/education, and then we have a classroom grant tab. All of the lessons and PowerPoints and student worksheets are available on our website for free. So if you're a teacher or if you need something to do with your kids on a vacation, you can go to our website and you can download all of those free activities and labs. And if you want to learn more about that, or if you want to donate to that program or our conservation program, you can go to this link. For this year, these are all of the nurseries that funded all of those teachers from all over the world. And Carnivorous, which was the logo in the middle, they sent free 3D printed carnivorous plants to every classroom teacher as well. And if you have access to a 3D printer, you can go to our website and you can print these super cool 3D models for free. Another way you can help support us is by going to our drop ship store. And Mathias, I don't know if you can see, but I'm wearing my favorite Nepenthes. It's the Nepenthes Cycle Carada with a little ant hanging on the thing. And everyone can get a tote bag, tank top, t-shirt, sweater, mug. Wherever you live in the world, you can order from this store, and they print locally, and we get all the proceeds, and all of the proceeds go to our education and conservation initiatives. Earlier, I mentioned that the ICPS is a volunteer organization, and we're trying to increase our voice in the world of conservation. And if you go to our Carnivorous Plant newsletter, you'll see that we have lots of different articles. If you're not a member, some of them are available for free, and you can learn about what we are doing to help carnivorous plants. And every other year we have a conference. This past May, we had a conference in Vienna, Austria. We do it every other year. So 2025, we're taking off, but in 2026, we're going to be somewhere in the Americas. So we might be in North America or Central America. We didn't have any bids for South America yet. All right, Mathias, I have a couple of questions from you, from the audience.
Okay.
Claudio asked, and this was at the beginning of your presentation, is it possible to tell if Nepenthes flowers have sepals and petals, Or is the corolla composed by sepals or petals, or are they considered tepals?
Yeah, the answer is they're considered tepals. There's only one ring of leaves around the actual sexual organs.
All right, Claudio also asked, is the genome of any Nepenthes species already fully sequenced?
Yes. So last year, the genome of Nepenthes gracilis was published. So fully assembled to chromosome scale. Yeah, including the sex chromosomes. And there was a project led by Kenji Fukushima, and I was lucky to collaborate on that.
Is there only one species fully sequenced?
Yeah, yes. Yes, I think so. Genomes of other species have been sequenced partially and not assembled. Yeah, so the assembly is the difficult part nowadays.
Okay, Rohani asked, what role might sex chromosomes play in the adaptability of Nepenthes in different environmental conditions?
Yeah, it could be that as I mentioned, that it could be that males and females actually have slightly different effective ecologies or physiologies, but that is really not well explored. So the sex chromosomes, because they're so the one species where we know for sure is Nepenthes gracilis, and in Nepenthes gracilis, the difference between the X and the Y chromosomes is really just the 3 genes. And it could be that these genes have functions that stretch out to the physiology of the plant, but it's more likely that that is indirect. So the males and females decide to flower with different frequencies and invest different amounts of resources, and that could lead to different. That leads to them experiencing the same environment in different ways. Yeah. But that's very speculative, so we need research in that direction, definitely.
Jackie and several other people say that was really fascinating. Thank you. And I think the last question we might have is from Rohani, who gets extra points for italicizing Nepenthes, the genus. They ask, could hybridization lead to unusual or disrupted sex chromosome configuration in Nepenthes that affect reproductive success or fertility?
Yeah, so if
I think a lot of times plants can hybridize and get crossed. They can get crossed a lot easier than a mammal or an animal.
Yeah, for sure. Yeah, there are lots of reasons for that. So definitely, in Nepenthes, we don't have this problem like when compared to animals where so in animals, it's sometimes the case that the crosses can be made only in one direction, and then yeah, either the male offspring or the female offspring of hybridization is not viable. But that doesn't seem to be the case in Nepenthes. But data is lacking, so somebody has to actually make hybrids in a controlled way, then raise them, and then say if the 2 directions of the crosses yield different frequencies of males and females, for example. It could be.
So maybe like 100 years ago, researchers decided that the mouse was going to be the model animal for a whole slew of things. Do you know if there was a reason for Nepenthes gracilis to be chosen?
Yeah, it was chosen because it's one of the smaller genomes, and it was so it's straightforward to grow actually. Yeah, it doesn't require as much space and time as other species, and this was also a collaboration with Singaporean researchers, and it's native to Singapore, so it was possible, just the logistics, to get fresh tissue from real wild Nepenthes. Yeah.
All right. Claudio asked, do all Nepenthes species share the same number of chromosomes, which you kind of just answered? What would be the species concept for Nepenthes if they can produce viable offspring when they hybridize?
Yeah, yeah, very good question. So the Nepenthes, all of the Nepenthes that have been looked at have 80 chromosomes in diploid normal cells or in the haploid cells. So in the pollen, there would be only 40 chromosomes. And that seems to be the case in all the species that have been investigated. So regarding the species concept, so yeah, people get training still to this day thinking about species concepts from the animal side. And in animals, hybridization is rare. Species boundaries are often very clear cut. But the closer one looks also in animals and in many plants, species boundaries are not of this nature. So species, if the idea is that some parts of the genome can be exchanged by hybridization and other parts of the genome not. So this is counterintuitive, so, but it means that hybridization only occurs effectively for some parts of the genome. So the so-called porous genome idea that is gaining track, and in Nepenthes that's definitely the case. So they hybridize, they have introgressed for probably hundreds of thousands, maybe millions of years, and nevertheless they are able to remain separate species. Yeah. So there's no argument against saying that Rafflesiana and Mirabilis are different species, but they still exchange some genes sometimes.
Yes, and then we go back to the old question or the old idea about clumping and lumping.
Mm-hmm. Yeah. Yeah, people have different views on this, but so so my take is if species coexist in the same space, so they have the opportunity to blend, but they didn't, then they are species. Then they're different species. Yeah.
All right. So thank you everyone for attending this webinar live. Thank you, Mathias. And we look forward to people attending our other webinars coming up. And I'll put your information in the bottom of the video so people can contact you if they're doing research. And thank you everyone for joining us and have a great day.
It's not a surprise that gardeners, educators, and scientists are fascinated by these unique plants. The International Carnivorous Plant Society, or ICPS, not only love these plants, but welcomes growers just getting started all the way through professional scientists. The ICPS even started an annual World Carnivorous Plant Day to celebrate them. The free online event is held the first Wednesday of May. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.